The question is, cell junctions of epithelial cells associated for rapid transfer of ions, small molecules and sometimes large molecules are called. Options: 1. Plasmodesmata, 2. Tight junctions, 3. Adhering junctions, 4. Gap junctions. So, here we are talking about cell junctions related to epithelial cells. When you look into the first option, plasmodesmata, plasmodesmata are the cell junctions are found in plant cells. Example, meristem. So, thereby the option 1 can't be an answer. When you talk about epithelial cells, the cell junctions in epithelial cells are called as desmosomes. And when you look into desmosomes, there are 3 types of desmosomes, and that is what is mentioned in the option 2, option 3 and option 4: tight junction, adhering junction and gap junction. Remember, tight junctions are non-leaky junctions. They are meant to prevent the leakage, and thereby option 2 is not an answer. Adhering junctions. Adhering junctions are cementing junctions, the one which is going to help in anchorage of the cell, attachment of the cell. But here they have to, junctions have to permit the transfer of ions of small molecules and sometimes large molecules. Thereby even option 3 is not an answer. So, it is the gap junctions that permit the movement of small and sometimes large molecules. So, thereby the answer is option 4. The question is, the non-staining secondary constrictions seen at a constant region in few chromosomes are called. Let us come to the options: kinetochores, satellites, nucleosomes, heterochromatin. One by one we can go to the options, there we discuss and arrive at the answer. When you talk about chromosome, a typical chromosome, if I take a simple sketch, if I have to draw, say this is the outline of a chromosome, and this is called centromere. Centromere is also called primary constriction, and these are chromosomal arms, also called chromatids. And centromere is the location where we have plate like structures called kinetochores, and they are going to have microtubules. So, kinetochore is plate like structure found in the centromere, and chromosomal arms are the one where chromatin is going to be present. It passes through the centromere like this, simple thread like coiled structures. So, here non-staining secondary constriction, primary constriction is centromere, which is going to carry the kinetochore. Thereby this is not an answer. Remember, kinetochores are associated with separation of chromosomes, as the duplicated chromosomes are held by a common centromere, and the centromere splitting is going to help in moving a part of replicated chromosome that happens during the anaphase. In case of mitosis, anaphase; in case of meiosis, anaphase 2. Fine. Now we come to the satellite. some chromosomes, in addition to primary centromere, are going to have another narrow area, what is called as secondary constriction. Secondary constriction areas are also called nucleolar organizing centers, and these are the sites where nucleolus is going to be produced, thereby nucleolar organizing center. The chromatin that passes through it is called nucleolar chromatin, that is the one which undergo transcription to give you ribosomal RNA. I hope all of you know nucleolus is a non-membranous structure present inside the nucleus where ribosomes are going to be manufactured or formed. And the part after the secondary constriction, this part is called as satellite, and such a chromosome is called as SAT chromosome. SAT refers to synacidothymidine, thymidilic acid is absent there, and these are the regions where the staining is going to be minimum, non-staining, and thereby the option 2 is the answer for us. Now we look into the other options, nucleosomes. In the 12th standard, in molecular basis of inheritance, you have spoken about nucleosomes with respect to eukaryotic chromosomes. Ultrastructural units of eukaryotic chromosomes are called as nucleosomes. Nucleosomes are made up of histone proteins and DNA. When you talk about histone proteins, it forms histone octamer. 4 proteins, I hope all of you know, H2A, H2B, H3, H4 in pairs constitute histone octamer. Over that, twice, actually 1.75 turn of the DNA winds over it, representing 200 base pairs. So now we know what is nucleosome also. Heterochromatin. When we are going to stain the chromosome of prophase, during prophase what is happening means chromatin undergoes condensation. Some of the regions are going to be more condensed and some regions are less condensed. When you are going to stain them, the regions which are more condensed are going to take deep stain, and we call such areas as heterochromatin areas. They are condensed means genetically inactive regions. The regions which are less condensed are open up areas. They are genetically active regions. We call such regions as euchromatin region. This property of prophase chromosome showing heterochromatin and euchromatin due to differential staining is called as heteropyknosis. So now you know the answer is satellite. We move on to the next question. The question is, the polymer of fructose is. Students, very carefully you have to remember this, because I have seen most of the time students are confused. It is very clearly given in the text in biological molecule chapter. So let me come to the options: cellulose, chitin, glycogen, inulin, the polymer of fructose. I hope all of you know homopolymer, the cellulose. Cellulose is a homopolymer of glucose. So thereby this is not our answer. Also remember, cellulose is the most abundant carbohydrate on earth. Then we come to the second one, chitin. Chitin is a heteropolymer. We know chitin is found as the part of exoskeleton of arthropods. Chitin is also a constituent of the cell wall of fungi, and it is the second most abundant carbohydrate actually on the earth, and it is a heteropolymer. It is made up of N-acetyl glucosamine. So, thereby this is also not the option, it is a heteropolymer. Glycogen. Glycogen is commonly called as animal starch. Again, glycogen is found as the reserve food material in animals including human, and it is also reserve food material in fungi. And it is a homopolymer of the glucose. Thereby, even this is not the correct answer. Now we come to the option 4, inulin. Please remember, inulin is a polymer of fructose. It is a sweetest sugar. Don't get confused with insulin. Insulin is a protein hormone secreted by beta cells of islet of Langerhans, which is called as hypoglycemic factor. So this is inulin, not insulin, and the answer is inulin. Now we move on to the next question. So the question is, during G1 phase of cell cycle, if the DNA is denoted by 2C and the number of chromosome 4, note down this, DNA is 2C, number of chromosome is 4, by the end of S phase, the DNA and number of chromosome would be 4 options are there. So when we look into the cell cycle, what we have studied, remember in the cell cycle we are going to have M phase, which is going to consume half an hour to 2 hours in 24 hours cell cycle. If you take 24 hours as one cell cycle, rest of it is taken by interphase. The interphase is going to include 3 important phases, namely G1 phase, S phase and G2 phase. So interphase is G1, S and G2. M is mitosis, where the cell is dividing. If the cell like nerve cell, muscle cell do not divide, we call then the cell is in G0 phase. And the question here is, at G1 phase, the DNA is 2C and chromosome number is 4. And remember, S is a synthesis phase during which DNA replication occurs. Suppose the chromosome is this and it has the DNA, and during S phase what happens? The replication of chromosome occurs and still they are held by the centromere. The replicated chromosomes are still held by the centromere, but the amount of DNA doubles. Thereby DNA is going to become 4C, chromosome number remains 4 itself, only this is the change. So DNA is 4C, chromosome number is 4. Let us look into the options. Option 1 says the DNA is 4C, chromosome is 4 respectively, thereby that is the answer. The other options can't be the answer. 2C 4, 4C 8, 2C 8. The question is, if a plant cell placed in a solution shrinks away with its cell membrane, it denotes that the cell sap has. So, what does it mean? The cell is shrinking when it is placed in a solution. That means there is going to be what is called exosmosis. Plant is losing water. And what is osmosis? Net movement of molecules of water from the region of higher water potential, that is greater water potential, to the region of lesser water potential. Higher water potential means less solutes. Maximum water potential is for pure solvent. By adding solute, potential is going to decrease. Cell sap can't be a pure solvent, and the solution must be having greater solute concentration compared to the cell. So, answer that we have to look is less solute concentration and greater water potential. Let us look into the options. More solute concentration, thereby it is not an answer. More solute concentration, again not an answer. Less solute concentration, yes. High water potential, yes. Thereby option 3 can be the right answer. Option 4, less solute concentration, correct. Low water potential, no. Thereby option 4 is wrong. So, dear friends, remember option 3 is the right answer for this question.
ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಭಾಗವಹಿಸಿದ ಎಲ್ಲ ಲೆಕ್ಚರರ್ಗಳಿಗೂ, ನಿರ್ದೇಶಕರುಗಳಿಗೂ, MCNDA ಸಂಸ್ಥೆಗೂ, ದೂರದರ್ಶನ ಚಂದನ ವಾಹಿನಿಗೂ ಹಾಗೂ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರಿಗೂ ನಮ್ಮ ನಮಸ್ಕಾರಗಳು.
We have a very interesting question, friends. Carefully look into it. Read the statements A and B. Select the correct option for the cause of appearance of brown spots surrounded by chlorotic veins in the plants. Now we come to the two statements A and B. Statement A, deficiency of iron, magnesium and calcium. Statement B, manganese toxicity. Quite interesting. We have to be very careful. When you talk about micronutrients, which are called trace elements, they are going to be required by less than 10 millimoles for the plant. We call them as micronutrients or trace elements. When the trace elements are going to be moderately less than what is required, it leads to deficiency with clear symptoms. If it is going to be moderately more than what is required, then it is going to be called as toxicity. So, here the statement A relates to deficiency, B relates to toxicity. When do you call any trace element has a toxic effect? Then the answer is very simple. If it is going to affect the growth of the plant, the dry weight, increase in dry weight by 10%, then we say it has caused the toxicity. Now with this, let us look into the options. Manganese toxicity, I am looking into the option 2. Manganese toxicity is going to affect the translocation of calcium to the shoot apex. So, here remember toxicity, it is extremely difficult to identify the symptoms, but manganese toxicity, when we talk about, it is going to affect the translocation of calcium to the shoot apex. So, it is causing deficiency of calcium because calcium is unable to reach the shoot apex. Second, it also known to cause deficiency of iron and magnesium. It competes with iron and magnesium for utilization. If its concentration is very high, naturally iron and magnesium are not going to be picked. That leads to that deficiency. In addition, manganese is going to bind to the enzyme site. It is going to be bound by magnesium. So as a result, manganese toxicity also lead to the deficiency of iron, magnesium and calcium, and thereby both option A and option B by itself can, or deficiency of iron, magnesium, calcium by itself can lead to this condition. So option 1 which says both A and B are correct. So this is the answer. Other 3 options are wrong. Now we come to a simple question. The greenhouse crops are option 1 bell pepper and sorghum, option 2 maize and tomato, option 3 tomato and bell pepper, option 4 maize and sugarcane. So dear students, when you have to answer this, you remember what you have studied in plant physiology related to respiration and photosynthesis. When you talk about carbon dioxide fixation by plants, in photosynthesis topic you have studied that there are going to be C3 plants and C4 plants. In case of C3 plant, the enzyme Rubisco is the one which is going to help in fixing the carbon dioxide, and this Rubisco enzyme is found in mesophyll cells of the leaf. In C4 plant, we are going to have 2 enzymes that are involved in carbon dioxide fixation, and one happen to be Rubisco and other one happen to be PEP carboxylase. I hope all of you remember it. Rubisco enzyme is found in the bundle sheath cells of C4 plant, where we talk about Kranz anatomy. I hope all of you remember that. PEP carboxylase is found in the mesophyll cells. Rubisco enzyme is the largest occurring enzyme on earth. Please remember that. And it is not only carboxylase enzyme, it is also the enzyme that is going to fix the oxygen, oxygenase enzyme. Suppose in C3 plant, if more oxygen is going to be available, then Rubisco is going to pick the oxygen instead of carbon dioxide, and this is going to affect the photosynthesis. So here what is going to happen is, at the cost of photosynthesis, neither ATP is generated nor the sugar, the glucose is going to be produced. And in case of C3 plant, thereby there is a need to provide more carbon dioxide. If more carbon dioxide is provided, then more photosynthesis occur because the oxygen concentration can be kept less. And what is the way, if we have to grow C3 plants of commercial importance like vegetable plants, then we opt for greenhouse. In the greenhouse, we provide more of carbon dioxide there. The carbon dioxide what is released is going to be again assimilated by the plant. So we have to look for an option of C3 plant, not C4 plant. Let us look into the option. Bell pepper is a C3 plant. So this is C3 plant. Sorghum, jola, it is C4 plant. So, this cannot be an option. Maize, again it is a C4 plant. Thereby, even option 2 is wrong. Tomato is C3. Tomato and bell pepper both are C3 plant. Thereby, the option 3 is the answer. They are the greenhouse crops. They are C3 plants. Maize and sorghum both are C4, and thereby that is not an answer. So, friends, remember option 3, that is tomato and bell pepper are greenhouse crops. For always remember it is C3 plants. We come to the question. The RQ value of tripalmitin is 0.7, then it is. It is a very simple question and easily you can answer. I hope all of you know the respiratory quotient is calculated as the volume of carbon dioxide evolved to the volume of oxygen consumed during the breakdown of respiratory substrate. I hope all of you know this. When we talk about the respiratory substrate, in case of carbohydrates, the RQ value is going to be 1, that is unit we call it. For organic acid, it is more than 1, like 1.33. For fatty acid and protein, it is going to be less than 1. For proteins, the RQ value is 0.9. I hope all of you remember in the text you have studied it. And for fatty acid, the RQ value is 0.7. So now look into it. If the RQ value of tripalmitin is 0.7, then it is naturally fatty acid. You can be asked anything and you may be given a reaction of breakdown of respiratory substrate, and in case of aerobic respiration, oxygen is going to be used, carbon dioxide is released. Calculate how much of oxygen is consumed and how much of carbon dioxide is evolved, and look into the value. If you are going to get 1, naturally it should be carbohydrate. If it is more than 1, organic acid 1.33. If it is going to be 0.7, fatty acid. If it is 0.9, protein. The question is, the plant growth regulator known to enhance sugar production in sugarcane crop is. It is just a knowledge based question. The options are cytokinin, auxin, gibberellic acid, ethylene. I hope all of you know plant growth regulators are 5 major kinds. 4 of them are mentioned. And one what is not given here is abscisic acid. Also remember, it was Charles Darwin who worked with his son Francis Darwin on canary grass first to talk about the possibility of the hormones or growth regulators involved in plants. And the first phytohormone to be discovered and isolated is auxin. Whenever we talk about auxin, we remember apical dominance. Auxins are extensively used in plant tissue culture to promote root formation. For vegetative propagation, if you have to induce the root formation, auxin is the best. And also remember, auxin was first, the name was given after isolation by F.W. Wendt, the famous Avena curvature test. But here the question is about enhancing the sugar production in sugarcane. Cytokinin, when we talk about cytokinin, auxin and gibberellic acid are called as growth promoters. Basically, we are going to classify the plant growth regulators into two kinds, growth promoters and growth retardants or inhibitors. Abscisic acid is a known growth inhibitor, whereas ethylene is both promoter as well as inhibitor. It has more a role as a promoter than an inhibitor. Cytokinin are going to be used to increase the shelf life of vegetables. Cytokinin also finds an use in plant tissue culture for shoot induction, what we call collagenesis. Cytokinin, I hope all of you remember the name kinetin and zeatin. There may be a question that where we may ask you which phytohormone or growth regulator is found in the liquid endosperm of coconut. The answer is cytokinin. And the first growth regulator to be isolated, discovered, the answer is auxin. At the same time, I want you to remember auxin and ethylene both are going to induce flower formation in pineapple, and also they are going to be used to prevent the germination or sprouting of the potato. So now we come to the gibberellic acid. The moment you remember gibberellic acid, I hope all of you remember, it was first extracted from a fungus called Gibberella fusicori. Wherever this fungus used to grow, the paddy crop in the paddy field, the paddy crop used to grow abnormally tall, thin, spindly, and we call that as Beccani disease, in English foolish seedling disease. Kurosawa was the one who showed that this fungus is associated with it. Yabuta and Sumiki isolated the gibberellic acid from the fungus. Now, large number of gibberellic acid are known to occur, which are going to be secreted by the plant tissues. And when we come to the question, enhance sugar production in sugarcane, it is gibberellin, that is gibberellic acid. Gibberellic acid play a very important role in internodal elongation. In case of sugarcane, internodal elongation means more of sugar. If we are going to use gibberellic acid, we can enhance the sugar production to the tune of 20 tons per acre. Naturally, the farmer is going to be benefited. So, the answer is gibberellic acid. In addition, I want you to remember, gibberellic acid is going to induce internodal elongation in rosette plants, and we call this phenomenon as bolting. The question is, select the factor that decreases the cardiac output. Direct, simple question. Options are, first one, adrenal medullary hormones. Option 2, neural signals from parasympathetic nerves. Option 3, sympathetic neural signals. Option 4, exercise. Dear friends, I hope all of you know what is cardiac output. Cardiac output refers to volume of blood ejected from left ventricle into aorta per minute. So it is minute volume of blood leaving the left ventricle into the aorta. And what is responsible for cardiac output? Naturally, it is the contraction of the heart chambers, what you call as heart rate. Increase in heart rate increases the cardiac output, because cardiac output is determined by two factors. One is heart rate, the second one is stroke volume. Stroke volume is like tidal volume. Here volume of blood entering into the aorta from left ventricle per ventricular systole contraction. So now let us look into the option which is responsible for increasing heart rate. In turn, it is going to increase the cardiac output, minute volume of blood entering into the aorta from left ventricle. Adrenal medullary hormones, they are nothing but catecholamines. They are namely epinephrine, norepinephrine, which is also called adrenaline, noradrenalines. My dear friends, all of you know these two hormones are called emergency hormones. These two hormones are called fight, fright and fear hormones. During emergency, it increases the cardiac output by increasing the heart rate. So thereby option 1 is incorrect. Now we come to neural signals and sympathetic, that is parasympathetic neural signals, sympathetic neural signals. Remember, human heart is a myogenic heart where the pacemaker is sinoatrial node, which is found at the base of opening of superior vena cava, and automaticity is the property of myogenic heart. What does it mean? Initiation of heartbeat, setting of basic rhythm of the heartbeat is taken care of by conduction system located within the heart, whose pacemaker happen to be cell. But heart rate can be increased or decreased, and we are going to have neural regulation, chemical regulation, which represent internal factors. External factor can be exercise. So if you are going to run a sprint of 100 meters, your heartbeat rate is going to increase. Naturally, cardiac output increases. Thereby, option 4 is also not an answer. Now, either it is 2 or 3. Remember, cardiac centers are located in medulla oblongata, acceleratory center, inhibitory center. The inhibitory center is going to start from medulla oblongata, passes through vagus nerve, innervate the sinoatrial node, and it is parasympathetic neural pathway. Whereas sympathetic neural pathway from the axillary center of medulla oblongata passes through spinal cord, and then it is going to come with the spinal nerve. That increases the heart rate to increase the cardiac output. So the answer is neural signals from parasympathetic nerves. This is the one which is going to decrease the heart rate. This is going to increase the heart rate. So even this is not the answer. So remember, the option 2 is the correct answer.